Before break, we had left off answering some questions about this alignment log file. So again, this is just a summary of the mapping information, so how the alignment went for the star alignment. And so we'll just continue on answering these questions. So I believe, Lisa, you said you were back. If you want to load up the next question, we are on, we're on this one here. Whereas were any splice regions identified, and if so, how many in total? So what is the total number of splice regions identified? And I'll give you guys a few seconds to answer that before we take a look at everyone's answer. I'm going to go ahead and share answers. So if you guys want to put in last guesses, if you're unsure, or if you're unsure, you could just say you don't know. So yes, a lot of yeses, a lot of right answers. Yeah, most of you are correct here, so I'll show you where to find at least those of you who answered. So those of you who haven't answered or didn't get it, if you go up here under the unique reads location, there's also a line here that says number of slices and gives you the total. It then, this is for the next question, it then breaks it down into number of annotated splice junctions and where those splices exist, and also the number of non-canonical or unannotated splice junctions that were identified, thanks to that two-pass mapping that we do. But so the total number of splice sites is given right here. So it is 38,550,564. Any questions about where to find that in this file for those of you that might not have gotten it? Okay, so we'll go ahead and answer the last question in this section, which is how many of those splice regions were annotated and how many were non-canonical or unannotated. And I'll give you guys a few seconds to answer that. So how many were annotated and how many were non-canonical or unannotated? Show. So we have, yeah, almost everyone got it right for those of you who answered. So let's go back over this. So the nice thing about this summary is it tells you this information. So if you look here, number of splices annotated, so this is going to be the number of splice sites that were annotated or that were already known. So it was already in the original index information in the original GTF file that was provided. And that number is going to be 38,550,311 is the number of splice sites that were identified in our sample data and the sample that we're looking at. And then the number of non-canonical ones are just below that. So it goes through and tells you the location of where those splice sites are. It does a breakdown for you for the annotated ones. And then it tells you the ones that were non-canonical or the ones that it found that weren't in a known location. And so these are the novel splice sites that we're able to identify and quantify because of the two-pass mapping that we set up to do. And so that's 14,147 were the total number of non-canonical splice sites. Anyone else not understanding how we got that? Okay, great. So similar to when we were going over the FASTQCs of our raw and our trimmed FASTQ files, it also will be pretty cumbersome to have to look at the alignment log files for all 12 of our samples. So we took just a look at one of our samples, our flight warp 1 sample here. And so we're going to revisit our multi-QC tool, which also is capable of identifying these log files from the star alignment tool and compiling all that information. So this should look pretty familiar to you at this point. So again, there's a link out to the multi-QC program. And now we're going to use multi-QC to compile all of the log final out files from each of our samples. So there should be 12 samples, 12 log files. And so again, we're going to call this multi-QC command. We're going to give it a prefix name, and this time, because this is the multi-QC of the align data, I put the prefix align underscore multi-QC. And then the -O option specifies where in our directory structure that we set up way at the beginning of this notebook to output that align multi-QC report. And then as a positional argument here at the end, we provided the directory to that log final out file, the directory to where all of the log final out files are for all samples. So we can go ahead and run that command if you haven't already. Is there anyone that's still waiting for it to run? Is anyone not seeing what I'm seeing on the screen? Okay, great. The standard output should look pretty familiar. Again, tells you the version, tells you if there's a new version, tells you which path it's looking in for the files to compile. It tells you now that it found 12 reports. Does that make sense that we're seeing 12? Yeah, why, Chief? You can unmute your mic or you can type it in the chat. Because that's the number of samples, I assume. Yeah, so that's the number of samples that we have. And keep in mind, when you do the star alignment, you align the forward and the reverse in pairs. So you come up with one set of output files for each sample. So just like what we saw in the BAM file, you see your forward and your reverse reads for each sample in the output file. So we should only see 12, which should match the number of samples. And then again, we see that the multi-QC is complete. We don't see any errors in here, so we know everything looked good. If anyone didn't see that, please let me know in the chat. There's construction back. Oh, that's fine. So again, here I list the parameter definitions to define what each one of the parameters in multi-QC is. The input data is the log final out from each of the samples that we're compiling. And then the output data, which you're used to by now, is a directory containing all the compiled summary alignment of the info and stats from each sample. And then all of that information in nice graphical format in an HTML report. And of course, you see the prefix that we gave it for both of these outputs, the align multi-QC. And similar to what it was for the FastQC, I have another link here that tells you how to interpret multi-QC data from, alignment log files. Lauren, it looks like Susie just joined us. Susie, if you could join Lauren in her breakout room so she can catch you up.
Will do, thank you.
Thanks. And then, Lauren, just let me know when you guys are back. And so to view the HTML report, again, which is a compilation of all of the log final out files from each of our samples, we're going to go ahead and run the Python library to view HTML files. So go ahead and run that if you haven't already, and then run this iframe command so we can open up that HTML report of our alignment data. It should look like this. Is there anyone who's not seeing this? Okay, great. So this is a compilation this time of the information in that log final out file for each sample. And so we should see only one line for each of our samples. As Chief pointed out, we should only have one of these per sample because the mate pairs are interpreted together in the alignment step. And so we see the percent of uniquely mapped reads, and we also see the raw number in millions, so the number of uniquely mapped reads in millions. And similar to the multi-QC with the FastQC data, we can sort these just by clicking them, which is going to help you guys answer some questions. And same with the millions. And as we scroll down here, some additional information we see is basically all the information that's in that log file, this time graphed out. And so this is set to the raw number of reads. I always like to look at this in terms of percentage because then it accounts for the total number of reads in each sample. It helps you compare samples a little bit more easily. And this is color coded, and it tells you the number of uniquely mapped reads, the number of reads that were mapped to multiple loci. the number of reads that were mapped to too many loci, meaning it went beyond the threshold of 20 that we gave it. Ones that the number of reads that weren't mapped because there weren't enough base pairs in the read that aligned, so unmapped because they were too short. And then we have a chunk here, which I don't really see many of these samples have, which is unmapped for a reason that isn't defined in the log file. And again, you can hover over each one of these. You see the samples listed on the left here, and it'll give you exact values in percentages, which is going to be useful in answering these questions. So again, compiles all this information in a nice way. When I look at these, I'm really looking to see, especially for well-annotated genomes like the mouse genome, I really want to see this number over 70% for uniquely mapped reads in the data. It just gives me confidence that things looked okay and nothing went badly. If I see less than 70%, I'll go back up and like look at the trim data and even the raw data and try to see if there's anything that makes sense as to why we're not getting uniquely mapped reads. So it might be less than that 70% threshold that I like to see it over because we have adapter dimers or we have contamination like ribosomal RNA that just takes up a lot of the reads and maps to ribosomal could map to multiple locations because of the highly repetitive regions in ribosomal RNA, so something like that. So if this isn't over 70%, again, for well-annotated genomes like the mouse, I'll go back and kind of look through some of the previous data and try to identify a reason for that. So pretty short, that's actually it, right? So this is all the information that it's graphing and comparing from the log file for each sample. So I want you guys to take a look at that multi-QC report and answer these questions. So Lisa, if you could cue this question up. It's the first one here is, what are the min and max % uniquely aligned reads for all samples? So the min and max % uniquely aligned reads, we'll answer that one first. I'll give you guys all a few seconds to answer that. Okay, I'm going to go ahead and show it. We have 80.2, 75.9, min-max problems, and did raw numbers. I think these are all right. Yeah, so great job to find that information. You would just scroll up to the top here and you could sort either on the raw numbers or on the percent. And so this is sorted from most % aligned reads to least. And so what we see here is there's 80.2% from flight rep 3 that were aligned, which was equivalent to 60.8 million reads for that sample. And the least % alignment was 75.9% from ground control rep 6, equivalent for that sample for 56.5 million. And so that's how you found that information. So again, really good alignment. This is the kind of alignment for that I'd like to see in a well-annotated genome like mouse. Last question for this section. What are the min and max % of aligned reads that map to multiple locations? So min and max % of aligned reads that map to multiple locations. Keeping in mind that that information is in here and you can hover over to see those values in the samples. So looking for percentage, min and max % of aligned reads that map to multiple locations. Again, you could hover over and the color coding is here. So go ahead and submit your answers for that question. Have a look to see what you guys put here. Eleven and 13, 11 and 13, 11 and 13. I don't know where these numbers came from. 11 and 13, 11, 13, and 75.9 and 80.2. So let's go over this. So we're looking for the min and max % of aligned reads that map to multiple location. So we're looking at this light blue area here. So this light blue area is going to indicate map to multiple loci or map to multiple location. And then you can kind of hover over and see, I'd like to see like which ones are the biggest chunks here. And so as you can see, this to know what has the most. So this is a pretty big chunk here, and you look at that, it's ground control rep 3, and it says it's 13%. If you notice here, mapped to multiple loci, and it's 13% of the total reads here. So I think the person that did the other percentages was looking at the uniquely mapped reads. I wanted the reads mapped to multiple loci. And so yeah, our ground control rep 3 and also ground control rep 4. This is rep 3, this is rep 4. They both have 13% of reads that were mapped to multiple locations. And then for the least number, you're looking for the smallest chunk of blue here, light blue here, I should say, which is going to be your one up from there, your flight rep 3, which again, hovering over, you could see these values. So your flight rep 3 sample is going to have 11% multi-map loci. And then if you go through and look at each one, that's actually the smallest percentage of multi-mapped reads. So that's how I found that information. Any questions on how to look at or interpret these log files from the star alignment step? Okay, awesome. And so now that we have our mapped reads, we want to quantify them. However, before we quantify them, we want to know, remember when I was talking about library prep and we went through how the Illumina library prep is a stranded kit? Well, we need to find out how that strand orientation was maintained relative to the reference. So before we quantitate, we need to know which sense or anti-sense mapped reads that we need to quantitate, again, based on the strandedness of the kit, if it's a stranded kit. And so in order to do that, the tool that we're going to use to assess strandedness needs the BAM files indexed. And so the aligned reads, we have to index them in order to allow the RCQC program to locate particular regions of interest. Oh, great. Lauren, how far did you guys get? Did you go through alignment multi-QC?
Yes, we finished the second question and we came back right when you were starting this next section, so it was perfect timing.
Perfect. Okay, great. So the tool that we're going to use again is part of this RCQC suite of tools to assess the strandedness of our data. And it needs these aligned reads, these BAM files indexed. And so in order for the program to locate particular regions of index or particular regions of interest in your BAM file, so it'll create an index for each one of these BAM files. And there's a program within the SAM tools that will allow you to index your BAM files. And so to learn more about the SAM Tools Index program, well, this is the SAM Tools program in general, which has a variety of different features. If you remember, in order to view our BAM files, we use the SAM Tools View function. So there's also a SAM Tools Index function, which allows you to index your BAM files. And so if you want to learn more about that, there's a link to that program. And so we're going to index all of our aligned sorted by coordinate out BAM files for all 12 samples using SAM tools by running this command. And so this is the SAM tools command. We're using the index function of the SAM tools command. This option is just to note the number of threads or CPUs that your machine can use to do this. So I allocated 2 when I did this, and then it's pretty easy. The only input it needs is a positional argument. So the last part of the command is just the input BAM files. And so we're setting this up as an array, and we have it set up using these curly brackets because that sample variable is going to change based on which sample it's running. And it's going to be indexing the genome aligned BAM files because the genome aligned BAM files are what is used by the RCQC infer experiment tool in order to assess strandedness, which we're going to get into in the lecture. And so again, the input data for this command is just your BAM files, your genome aligned BAM files for each sample. And those were the outputs from that star alignment step. And I want you guys all, and the output data is going to be an index file for each of your BAM files. So it basically has the same exact name as your BAM file, it just ends in .bai. And so I want everyone to go ahead and submit this Slurm script to our simulated job scheduler here. And you should see that your job was submitted. And then we're going to hop into the lecture and talk about strandedness. So was there anyone who wasn't able to submit their job to the simulated scheduler? Okay, let's talk a little bit about strandedness. So before we go there, I want to remind you all where we are in our pipeline. And so this is the end of aligned reads. And so we went ahead and we assessed the quality of our raw reads. We trimmed and filtered them to remove low quality reads or low quality base calls and also to get rid of adapters. We then did another assessment of the quality after trimming to make sure everything looked good. We then, well, I told you about creating a star genome index, and then we use that star index as well as our trimmed reads to align those trimmed reads to our reference genome. We then looked at the quality of the alignment, so how the alignment went using multi-QC compilation of the aligned log final out files of the alignment for each sample. And so we are now at this step where we are going to evaluate the strandedness of our data. And before, well, let's talk about it. Okay. So RNA can be transcribed from the antisense DNA strand to make sense RNA. And that's the RNA that can code for proteins. And so that looks like this, right? We have our DNA here, and we see the antisense strand of our DNA, which is in the 3' to 5' directed, can be transcribed in the 5' to 3' direction to make a sense RNA transcript. And those are transcripts that can be coding transcripts. But RNA can also be transcribed from the sense strand to make antisense RNA or non-coding RNA. So it can also be encoded by the sense strand, and again, you'll end up with an RNA transcript that's in the antisense direction. And so if we have a read that aligns to a region where a sense RNA transcript and an antisense RNA transcript overlap, like what's shown here. So this read we have here, it could have been derived from the sense strand or from the antisense strand. So how do we know the transcript of origin? And can we know? How do we know, how can we possibly know the transcript of origin? If we have this sequence here, this insert or template that aligns to this region of the reference genome, but we know that that region of the reference genome encodes for either a sense RNA transcript or an antisense transcript. So if we have this read that maps to this location, how do we know which RNA transcript it came from? Can we know? You guys can unmute your mics, type it in the chat. No guesses? Didn't we sequence the forward strand first? We sequence in the forward direction first. Remember, our templates are just chopped up RNA, right? So we have a sample, we extract RNA from that sample, and then we remove ribosomal RNA from there, and then we chop up that RNA, and then we convert it into cDNA. And so how can we know if we have a transcript then after we sequence it and then we do the mapping and it maps to both of these locations, and we know that a transcript can be derived from that location in the sense direction, the antisense direction, how do we know which one it came from? Okay, move forward. So prior to sequencing, cDNA fragments are enriched. If both strands of the cDNA fragments are enriched, read orientation would not be preserved. And so if you guys remember back to library prep, it didn't look like this. It looked like this one was stopping. So this is generating that second strand. Let me actually go back up here. So let's go back up here. And so here, if you remember from library preparation, again, took our total RNA, removed ribosomal RNA, chopped it up into small pieces, and then we converted those pieces. Again, this is the original orientation of the strands here. We made a copy, right? Our first strand cDNA, made a copy. And then we made the second copy of our cDNA here, right? And so if we were, and then we did the polyadenylation and added on our adapters. But then what we do is we have this last amplification step. And so this one, which was derived from our original RNA, right? So our original strand. And so we know that this is the reverse complement of that original strand, right? Because we made a copy here. This is a first strand, and this was this is the original strand, and this was the first strand of cDNA that we made. So right now it's reverse orientation from the original strand that it came from. And then we have to make a second strand, right? Because we have to make sure that this is double-stranded cDNA, if you will. But when we made our second strand of cDNA, we actually added U's here, which is going to come into play. But if we wouldn't have added U's, when we went ahead after we added our adapters, if we went ahead and amplified both strands in this final amplification step, we would have amplified the strand in the orientation reverse complement from the original, and also an orientation that matches the original. And so if we would have amplified both of these strands, hop back down now. So if we would have amplified both of these strands in both directions at that stage, we would not have been able to tell where it came from, because we didn't preserve, in this example, we wouldn't have preserved the orientation of the original strand, right? Because then we would have sequenced both the strand that was made from the original strand and the reverse complement of that, and we wouldn't have known which was which. And so if that were the case, and there are library prep methods that do it this way, and those are called unstranded library prep methods. So for that, for reads that align to a region where a sense and an RNA transcript and an antisense RNA transcript overlap, we wouldn't be able to know. So again, had we amplified the cDNA in both directions, we wouldn't have maintained or known the orientation of the original strand. So with stranded kits, what they do, like what we went over when we looked at library preparation, is when it made that second strand of cDNA, the second strand synthesis, it actually incorporated uracil instead of thymine. And then when it does the final amplification step, it uses a polymerase that can't make a copy past a uracil. So as soon as it hits a uracil, the polymerase stops working, it falls off. And so when we do the amplification step, that's where we're preserving the strandedness, because we are only amplifying one direction of our original copy. So now we've preserved the orientation of the templates that we sequence. So now we can know that we always kept the orientation of our original transcript. And so preserving that strand information during sequencing is going to help resolve read ambiguity in those overlapping transcripts transcribed from opposite DNA strands. So now if we preserve the orientation of our original RNA, then we can assess the orientation of those reads relative to the reference transcripts, and we'll be able to know the transcript of origin for reads that align to a region where a sense and an antisense RNA transcript overlap. So in this example, for this read, if we know that we preserved, so we had a stranded kit and we're able to preserve the orientation of our original RNA, And if we know that we preserved it in the sense manner, so in the same direction, then we'll be able to tell that this read here that could map to either the sense RNA transcript or this antisense RNA transcript, because again, these regions overlap, we'll know that it came from here because we preserved orientation of our original samples. Does that make sense? Is that not clear to anyone? Okay, great. So to properly quantitate aligned reads, we're going to need to know if the libraries were unstranded or stranded, right? And then if they were stranded, we need to know if the read orientation, what that read orientation is relative to the reference. So how is that read orientation preserved? So depending on if you preserved the first copy of cDNA or if you preserved the second copy of cDNA orientation, it could be the opposite relative to your reference. And so we need to know what orientation was preserved. And the information should be in the library kit document. So the library kit document should indicate if the kit is stranded or unstranded. And if it is stranded, it should indicate which direction it maintains the strandedness. However, as we found and came across, actually, we used to not do this in GeneLab and just relied on the kit documentation or what was in published papers. And it turns out that the strandedness info may be difficult to find. And also, if you mix and match library prep kits and adapters like we do, you could change the strandedness orientation. And also, we've run into issues where the strandedness information is incorrectly reported or not reported at all in scientific papers. So because of all these issues, we really needed a foolproof way to know the orientation of the RNA inserts that we're looking at. And so thankfully, there's this tool suite called RCQC, which stands for RNAseq Quality Control Package. And there's this module in that tool suite called Infer Experiment that will look at the orientation of your reads and the relative to the orientation of the reference. And it'll tell you which direction your stranded reads are, if they're in the same direction or if they're in the reverse direction relative to the reference. And so this kind of summarizes, so RCQC Infer Experiment compares the strandedness of the reads with the strandedness of the transcripts. And read strandedness is going to be determined from the alignment data, right? Because those genome aligned BAM files tells us the orientation of your inserts, of your templates relative to the reference. And so transcript strandedness is going to be determined from the reference transcripts. And I do want to note that in order for this tool to work, the reference needs to be in a specific format called a browser extendable data format or BED format. So we're going to go over what that format is quickly. So again, the RCQC Infer Experiment module requires that the reference that you're using to assess the strandedness of your reads needs to be in BED format. So we actually use this two-step process to generate a BED formatted reference. And so this starts with our GTF file. So our gene annotation file, if you remember, had the start and end location of references. And also, let's actually take a look at that. So if you remember our GTF file here, and keep in mind when you sequence, when you have a reference genome, so number 7 here, when you have a reference genome, it was just a sequence that was put together to create your reference. And so it could have, your sequence that was used to generate your reference could have been derived from the sense or the antisense DNA strand. And so in your GTF files, it tells you if it was from your forward or your reverse strand in this column, column 7. So that information is in the GTF file, so it tells you the orientation of your reference. And so in this case, all of the features that are listed here are on the reverse strand or the anti-strand of the DNA. So knowing that information, rather than it being in that format though, in that GTF format, which is here, This tool requires that information to be in a specific format known as a bed file. Yeah. And so we do a two-step process to go from the GTF file to the bed file. So we actually first make another file type called the GeneFred or gene predictions table from the GTF file using a program from UCSC called GTF to GeneFred. And so this is what a GeneFred table looks like. Again, this is our intermediate to go from a GTF file into a bed file. And again, it's just the format, it's how the information for annotated genes are shown. So in this type of file, you'll see the same information that we saw in the GTF file, just in a different orientation. So here I have all of the columns listed. So again, this is a gene predictions table with this just information from the GTF file in a certain format. So in this table, we have our transcript ID in the first column. We have the chromosome name in the second column. We have the strand orientation, sense or antisense in the third column. And then we have the start position, transcription start position in the fourth, transcription end position in the fifth. The 6th is coding start region. The 7th is a coding end region. The 8th is a number of exons that can be found in the transcript. So each one of these represent a transcript. Number 9, column 9 is the exon start position. So you should see the same number of start positions as you do number of exons. And then 10 is the exon end positions. So again, all this information is found in the GTF file. It's just in different columns. So it's just basically all that this program does is it reorganizes the information in the GTF file to put it in this orientation. And then we use another program called GeneFred to Bed, again from UCSC, which takes that GeneFred table and just rearranges things again into the bed format, which is ultimately what we need for this tool. So again, you should see similar information. There are some things added here. But we'll go through these. So the first column is the chromosome name. The second column now is the chromosome start or transcription start position. The third column is a transcription end position. The fourth column is going to be the name of your transcript. The fifth column, this is going to be associated with that genome browser track line. So if you guys remember just taking a look at this genome browser information, so this uses a bed file to identify regions of interest in your reference. And so a bed file is actually what's being shown in here. And so this uses that. So in that bed file, information specifically for a genome browser is in there. So that's what that's for, is associated with those track lines. And then number 6 here is going to be strand orientation. So again, sense or antisense. 7 is going to be the coding region start site or exon start site. 8 is going to be the coding region end or exon end site. Number 9 is again associated with the genome browser. It has to do with the color that's being shown for that region of the transcript. Number 10 is the number of exons. And number 11 is the exon end position. And number 12 is that exon start. So again, same information really that's in the GTF file and ultimately in the GeneFred table, just in a different format. So these tools were developed at different times by different people, and so that's why they don't all start with that same input file for the reference. So we use these other tools that were created to convert these files, basically just rearranging information around about our reference. So this is a file format, the reference file format that's needed for the RNAseq QC and for experiment module to determine strandedness. So how this works is we run this package of RNAseq QC called infer experiment, and .py means that it was built in Python, so it's a Python program. It requires a couple of parameters that we add here. So you'll see a -r, which is going to specify the path to the reference gene model in bed format. So reference information in that format that I just showed you. The -i option is going to specify the path to our input BAM file. So those are the aligned to genome files that were outputted from STAR for each sample. And then this -S option is going to specify the number of reads to sample from the input BAM file. And so basically, it doesn't necessarily have to look at every single read in your BAM file to get an idea of what the strandedness orientation is. And so you can look at every single read in your file. But really, if you look at 30% or so of your reads, you should be pretty confident in whether or not your data are stranded, and if they are stranded, which orientation it is relative to the reference. And so here's an example of some of the outputs. So this is all printed right in the standard output, actually, of the infer experiment. This is an example for single end data that's non-strand specific. So remember, the data that we've been working with has been paired end, which means that we sequence those templates in both directions, in the forward direction and the reverse direction. You can also just sequence in the forward direction, which some people do. And this program will take either or. So if you give single end data into it, the output looks like this. So this is what the output looks like for an unstranded kit. So what we see here is about half the reads. So this is read orientation of the reference compared to the read. And so basically, anytime we have a reference in the positive orientation, a read is in the positive orientation. Every time we have a reference in the minus orientation, the read is also in the minus orientation. So basically, if these two match, it means it's in the same orientation relative to the reference. If these mismatch, so when the reference is in the positive direction, the read is in the negative direction. If the reference is in the negative direction, the read is in the positive direction. So here it's just saying that the read and the reference are in opposite directions. And you'll notice half of the reads that were sampled in this example were in the same orientation of the reference. The other half of the reads that were sampled, roughly half, the orientation of the read was in the opposite direction of the reference. And so this is what you would see for an unstranded single end data. So about half the reads same orientation, the other half the reads opposite orientation relative to the reference, and there's always going to be some percentage of reads that will probably fail to be able to determine the strandedness, maybe just based on the read size, and it just happens to match exactly in both orientations relative to the reference. So like palindromic sequences and things like that, you won't be able to know the read orientation. If you have strand-specific single-end data, what you'll see is something like this. So here we have, if you look at this number here, this is the fraction of the reads that are in the same orientation of the reference. So again, if the reference is in the positive direction, the read is also in the positive direction. If the reference is in the negative direction, the read is also in the negative direction. And we see that almost all the reads are in the same direction as the reference. Down here, this is describing again that when the reference is in the positive direction, the reads are in the opposite. When the reference is in the negative direction, reads are in the positive direction, which means that the reads are in the opposite orientation of our reference, and that makes up a small fraction in this case. So this is what it looks like when there is single end data that's strand specific. So in this case, this is stranded, and it's stranded in the sense orientation, again, relative to the reference. So whenever I say sense or anti-sense, I'm always saying relative to the reference, because as we saw in that GTF file and also in the bed file, your references can be in the sense or anti-sense direction, depending on how the genome was built and how it was sequenced. So whenever I use sense and anti-sense, it's always with respect to the direction of the reference. So this is sense or the same orientation, the reads relative to the reference. And most of them are in that one direction. And so this is why this set of data would be considered stranded and stranded in the same orientation relative to the reference. Now we're going to see what this output looks like for paired end data. So for paired end data, you're going to see a bit more information because we have two reads, right? We have a forward and a reverse read for each template. And so here, the one indicates the forward read. So here we have the forward read is when the reference is in the positive orientation, the read is in the positive orientation. Again, for the forward read, when the reference is in the negative orientation, the read is in the negative orientation, and that's going to be the opposite for the reverse read, right? Because remember, mate pairs are always reading in opposite directions of your template. And so if this is the case, where your forward read is in the same orientation with respect to your reference, then of course the reverse read, indicated here by a 2, should be in the opposite orientation relative to your reference, and we see that it makes up about half. And then down here, this is the fraction explained by the forward read being in the opposite orientation relative to the reference. So here for the forward read, indicated by the 1, we see the reference is in the positive direction and the read is in the negative direction. And here in the 2, and so anytime the forward read, for the forward read, anytime the reference is in the negative direction, the forward read is in the positive or opposite direction of the reference. And the reverse read, indicated here by a 2, is in the same direction relative to the reference. And so here, the reverse read, anytime the reference is in the positive direction, so is the reverse read. Anytime the reference is in the negative direction, so is the reverse read. And so again, you see this is about half and half. And so this is an example of what this output looks like for paired end data that's from an unstranded kit. Because again, we have about half of the reads where the forward is in the same direction as a template, and then of course the reverse is in the opposite direction of the template. And about the other half of the reads that were sampled here has the forward read in the opposite orientation of the reference and the reverse read in the same orientation of the reference. So these templates overall are going to be, for this one down here, it's going to be in the opposite orientation of the reference. These templates overall, if we're looking from forward to reverse, is going to be in the same orientation of the reference. This is what strand-specific output looks like for paired end data. So again, we're looking here at the fraction of the reads that are explained whenever you have the forward read here. If the reference is in the positive direction, the forward read's in the positive direction. If the reference is in the negative, then the forward read is also in the negative direction. And then of course your reverse reads are going to be opposite. So in this case, your reverse read, if the reference is in the positive direction, your reverse read would be in the opposite or negative direction. And anytime your reference in the negative direction, your reverse read would be in the positive direction. So you see that that's a very small fraction in this example. And then if we look at the fraction of reads explained by this being that the forward read here is going to be in the opposite orientation of the reference, and the reverse read is going to be in the same orientation of the reference. So here again, for your forward read, anytime the reference is in the positive direction, the forward read is in the negative direction. Anytime the reference is in the negative direction, the forward read is in the positive direction. And then you see the opposite for your reverse read. So anytime your reverse read for the reference in the positive direction, your reverse read is also in the positive direction. Here, anytime your reference is in the negative direction, your reverse read is also in the negative direction. So in this case, we have most of the forward reads are in the opposite orientation relative to the reference, and most of the reverse reads are in the same orientation relative to the reference. So this is also stranded, right? Stranded because one of these is dominating, and the orientation in this case is going to be antisense. Why? Because the bulk of the fraction of reads that were sampled here, the forward read is in the opposite orientation of the reference, and your reverse read is in the same orientation of your reference. And so these templates are going to map to the reference, but if you look at your forward and your reverse read, it's going to be the reverse orientation compared to your reference. So this is another way to look at strandedness, and this is antisense, again, with respect to your reference. Takes a lot to get your head wrapped around strandedness. Any questions about that before we do it in the Jupyter notebooks? You guys feeling good about that? Okay, let's do it. So everyone can go ahead and jump back to their Jupyter notebooks. And you guys all should see it say Python conda environment colon GL4U idle. If anyone does not see that, please let me know. Okay. So our next step 5 here, is going to be to create that bed file for the reference genome. Again, similar to the star index that we only had to make once, so long as we have the same read length. For the bed file, we also only have to make this once, and then we could use that bed file for all of our samples, all of us can. So I already made this for us. And so just as a quick review here, prior to quantitating our alignment data, we're going to have to determine if and how the reads were stranded during library prep and sequencing. And to do this, we're going to compare the strandedness of the reads with the strandedness of our reference transcripts. And we're going to do it using the RNAseq QC infer experiment program. And just a note here, the strandedness of reads is determined from the alignment data, and the strandedness of the transcripts is going to be determined from the reference genome annotation, so that we've converted to a bed file. So since our samples came from the soleus muscle of mice, we're going to use the GTF file associated with mus musculus, so the same GTF file that we used when generating the star reference. And again, just another link here to that ensemble database that we went over where you can find that GTF file. However, the RCQC for experiment programs, as I mentioned, requires those annotated transcripts to be in a specific format, known as a browser extendable format or bed format. And so before we can assess that read strandedness, we first have to convert that GTF file to a bed file. And we do that in GeneLab using this two-step process. Step 1 is converting the GTF file to a GeneFred table format that we just went over. And that uses this GTF to GeneFred tool. And to learn more information about that, you could click on the link. Step 2 is then going to be to convert that GeneFred table into the bed format using the GeneFred to bed tool, which you can click on there to learn more information. And if you're curious, want more information about the table format, I also have links here to the GeneFred table format and also the bed file format if you want to look up more about the format that we went over. And then again, as a little note here, this only needs to be done once, so we already did it for you. And the following commands were those that were used. So we went over these in the lecture. Super simple. This GTF to GeneFred just needs a GTF file as an input, and these are all positional arguments, so it expects that GTF file to be first. And then the end file, you can give it a file name of what you want the output GeneFred table to be named. So I name it the exact same name as the GTF file, but then I do a .GeneFred to let me know that this GeneFred table came from that GTF file. And then just going over the definition of those two positional arguments here. And then step 2 is then to create that take that output GeneFred file and use that as input to the GeneFred to bed command. And that would give us an output of a bed file. And then again, you can give it whatever name you want. I keep the name again consistent with the GTF file that all this information originated from, except this time I use the extension .bed to let me know that this is a bed format of that information. Going over these parameter definitions again. This parameter definition is not in there, so I need to remove that, so you guys can ignore that. And it just needs that GTF file, and it's the input at the start, and then the GeneFred file is going to be the input for the second step of the two-step process. So ultimately, your input data is going to be that GTF file for us from Ensemble. And your output data is going to be the GeneFred file from the first step, and then ultimately the bed file from the second step. And the bed file is what we're going to use to determine read strandedness. Any questions about those reference files? I know it's annoying that we have to change the formatting of this information depending on which tool we use. Okay. So let's go ahead and evaluate our strandedness now that we have our reference in the correct file format. So again, just a reminder why this is important to know. the strandedness prior to quantitation. Stranded RNA sequencing preserves strand information of a read, which is going to help us resolve read ambiguity in overlapping transcripts transcribed from opposite DNA strands. So again, help us with those reads that could map to an antisense or a sense RNA that both come from an overlapping region of the genome, and so they'll have the same sequence just in the reverse complement. So this information is going to be required in our quantitation tool that we're going to be using RSEM in order to properly assign reads to the correct gene of origin. So to do this, we will compare the strandedness of the reads with the strandedness of the transcripts using that RSeq-QC infer experiment program. Just a little note here that RSeq-QC is a tool suite. So it's a whole package. It contains several programs used to assess the quality of RNA-seq data. So just so you guys know, the current pipeline of our current RNA-seq pipeline actually uses a few other packages. And so what you'll notice in our repository ever since we updated our pipeline is that we also assess our alignment data using the RSeq-QC gene body coverage analysis, which looks at the read coverage over the length of transcripts. We also do a read distribution, and this is to quantify how the align reads distribute over genome features. And we use, for paired end data, we use this inner distance tool, which is used to determine the insert size between two paired end reads. So distance from the end of our run to the start of R2. So a little different than the P10 length that we have outputted from the BAM file. So just so you know, we do use those additional QC. We thought this is already so much and you don't actually need this information to process the data. So we're not going to go through and do those during the bootcamp, but I did want to let you know that we do that normally for our data on GeneLab. And if you want to learn more about any of these tools, I gave you links for all of them. Okay, so we're going to evaluate read strandedness using the following command. This too, it doesn't take too long, but it takes at least a few minutes depending on how many reads you sample. And so we're going to go ahead and submit this to our Slurm job scheduler, again, as an array script, because we have to do this for every single sample. And so this is just the command that I went over in the lecture. So we call the infer experiment program using infer_experiment.py. -R is going to be the option to indicate where the reference is. And again, that reference has to be in bed format, which is what we just made, or at least I showed you how I made it just above this. We have this -I option, which is going to indicate where the input BAM files are. So again, you'll notice the curly brackets around sample because we're submitting this as a Slurm array script. And so that sample variable is going to change depending on which sample is being analyzed. And then we have this -S command, which is the sample size. So the number of reads to sample. And at GeneLab, we kind of played around with a few different numbers here, and we realized that for our case and purposes, sampling 15 million reads is enough for us to be confident in the strandedness. So we played with this number a little bit and then kind of decided based on resource usage, how long it takes and what additional value we're getting. And so we settled on 15 million reads. So we always sample 15 million reads of our data. And as I mentioned, the outputs that we went over in the lecture just print out to standard out. I kind of want it as a separate output file. And so what I do here is, if you guys remember from the intro to Unix, when we were creating new files, we would use this caret to pipe the output of one command to a new file. So that's what we're doing here. So essentially the standard output, we're just gonna pipe it into a new file. And I just labeled that file. This is in the directory structure we set up at the beginning, in that RSeq-QC analysis subdirectory, in the infer experiment subdirectory, and just naming each one of those files as sample name _ infer experiment .out. And so again, just defining all those parameter definitions here. And then our input data, two pieces of input data. We need our reference bed file, and then we need the genome aligned BAM file. And so that was outputted from the star alignment step. And then what we're going to get out is, of course, the file that we push the standard out to, which is this infer_experiment out file. And that's going to be a quantitation of the read strandedness relative to the strandedness of the reference transcripts. And so I want everyone to go ahead and submit this Slurm job to our simulated job scheduler. So go ahead and run that. And you should see submitted batch job with a job ID. Is there anyone who's not seeing that? Okay. And then we just have 2 minutes left, so we're ending for the day. So let's open this up, but we'll probably wait until tomorrow to go over it. So you guys can go ahead and run this. And this is looking at for that first sample, the first biological replicate of our flight group. We're looking at flight rep 1, that output for inferior experiment.out. And then we see here what the output looks like. Again, this is paired end data, so it knows that it's paired end data, right? We gave it, that information is in the BAM file, so it can interpret if this is paired end data or not. So it tells you, hey, I read this, it's paired end data. And again, it always gives you some fraction of reads where it just can't determine the strandedness of. And then it'll tell you the fraction of reads explained by this orientation. Do you guys remember from the lecture, is this going to be the insert or the template that we sequenced? Does that indicate the same orientation as the reference or the reverse orientation of the reference? You guys put your answers in the chat. Yeah, you guys got it. So this indicates that for the forward read, the orientation of the reference is, if that's in a positive direction, the orientation of the forward read also in the positive direction. When the orientation of the reference is in the negative direction, the orientation of the forward read is also in the negative direction. And of course, it's always going to be the opposite for the reverse read. And so in this case, because the forward read is in the same orientation as the reference, that means the reverse read will be in the opposite orientation. So when the reference is positive, the reverse read is going to be in the negative direction. When the reference is negative, the reverse read is going to be in the positive direction. And we see a very small amount making up the fraction that is in the same orientation of the reference. And then if that is the same orientation of the reference, then what does this represent? Templates that are in the same or reverse orientation relative to the reference. Go ahead and put your hand. Yeah, exactly, Alba. Reverse orientation. So this indicates reverse. And you'll notice here, just a little hint before we end for the day, that we see the majority of the reads seem to be falling on there. So when we start tomorrow, we'll quickly review strandedness, and then we will answer some questions about the strandedness of our data. So it is 3 o'clock. I do want to let you guys know, if you want to stay on and play around with this a little more, if you want to rerun any of it above, you're more than welcome to. Just to give you a heads up where we are in the pipeline here before we sign off. You just go all the way to the top. We right now are in this step here, so we just evaluated strandedness. And so we're going to take a look, compile that information again with multi-QC first thing tomorrow, and take a look at the strandedness of our data. And then we'll be moving on to quantitation, which is the last step in this Jupyter notebook. So when you guys are done for the day, again, as a reminder, file and at least either do hub control panel and stop your server, or you can just go ahead and do file log out. So either one of those is sufficient to stop the server.